Okay, so let's get started on this lecture 16 of bio microelectromechanical systems. Quickly doing a brief review of the last lecture. So we talked about introduction to DNA structure, described the double helical structure, the Watson-Crick model of DNA in details, how the base pairs are linked to one another using different hydrogen bonds, and that makes the base pairs quite complementary. We also talked about aspects related to DNA hybridization, and this used as a modality for detection of sequences, rapid detection of sequences on the DNA strands. So you have to have a kind of capture probe and a library of these probes for being able to detect certain sequences which could be potentially on the targets or the samples of interest. We talked about stringency in the hybridization process, how stringent hybridizations or reduced stringency would cause a reduction in the specificity of detection. We do discuss this totally new concept of how chemical signal can be amplified by copying a certain sequence in a molecular reaction n number of times using what we call the polymerase chain reaction. Okay, it is one of the breakthrough technologies of this decade, which has necessitated the huge, or which has essentially been the foundation of the huge molecular diagnostic industry. We also talked about how to design certain steps of this reaction and where the reaction can be rate limiting, or which step of the reaction could be critical to limiting the rate of the reaction. Now today we will go ahead and do how, once the DNA has been obtained, how we really obtain this information about whether it is being copied or the sequences that we are wanting to is being copied or not. In other words, we need a mechanism to find out whether we can on a real time basis detect snapshot by snapshot of different stages that this reaction is in as the DNA copies and becomes multiple folds. So because it is just molecules and they cannot be just visualized, we need to rely on spectral information. It could be an absorption spectra, it could be a fluorescence based spectra. So typically for detection we do need something which would be a high signal detection technique, and therefore fluorescence in most of the cases preferred for detecting the growing DNA strands along the PCR reaction. This process is also known as the qPCR, quantitative PCR, or the real-time PCR. So let us see what happens, what are the techniques, different techniques where we can club the fluorescent dye to the DNA molecule, and also see how a growth would be indicative of what stage the reaction is in. So the first technique that is very useful is called a cybergreen assay. Cybergreen is a fluorophore. It is essentially a very high quantum yield fluorophore, and these really are different for RNA and DNA. The way they bind to RNA may be totally separate than the way cybergreen binds to DNA. These chemicals are available and have to be really stored in oil based emulsions for a better lifetime or a better quantum yield or shelf life of this particular fluorophore dye. The basic operating principle is very simple. It works on the principle of intercalation. Okay, I explained last time what intercalation really means. So if you have a DNA structure which is more like a cage like form, okay, so you have this helical strands, two helical strands of DNA with different base pairs, and then this fluorophore molecule goes and sits without really covalently bonding into this particular cage like structure, and this results in a change in the orbital configuration and makes or attains an overall state where fluorescence is possible. So if in that case there is an excitation frequency of, let's say, a certain lower wavelength, it results in an emission frequency in the green region for particularly cybergreen. And the idea is that if these copies of DNA molecules grow, as they grow along along a successful PCR reaction, we should be able to get an increase in the green fluorescence, which makes sense because more the number of copies are, more is the intercalation level of the dye is actually available in plenty in the reaction. I should be able to get a snapshot of what is the overall concentration at a certain point of time in the reaction. Okay, there is another assay which is very commonly used, and so there is a binding limitation, okay, in the cybergreen assay. And let me just reiterate this again, that this is just by virtue of intercalation that the signal comes out. So it is almost always obvious that irrespective of whatever be the sequence of the DNA, or irrespective of whatever be the nature of the DNA, you should still get a signal, because the moment it is a double stranded structure, a cage like structure, this dye gets in and starts fluorescing. Okay. Now in a PCR process, as we know that there are about 30-40 cycles of different thermal steps. Okay. So you have to take this reaction through a step of 95, through a step of again about 50 degrees, and then another step of 72 degrees. So due to this rapid thermal cycling, there is always a tendency of the junk molecules to go away with time. Essentially, if it denatures or it renatures, it all is jumbled up, and beyond a certain time it starts really disintegrating, because there are no specific end recognition sequences to pick up the junk. Okay. However, the end recognition sequences do pick up the target DNA and amplify it many folds, let's say about 1 billion copies. Okay. And the success or failure of the cybergreen based detection technique really bases itself on the statistically significant number. Okay. So if you're talking of a comparison of 1 billion molecules to molecules of a few tens or a few hundreds of molecules, then it doesn't make any sense to compare, because the signal in case of 1 billion molecule is going to be many folds over the trace amount of junk molecules which are available. So the cleanliness of a PCR process in terms of what you are amplifying or what is remaining back after this rigorous thermal cycling step, in combination with a protocol like intercalation, might make a difference and might give you a good estimate of the fluorescence growth. The second very useful technique which is used is, and just to suffice this condition, this necessary condition of non-specificity developed by techniques like intercalation, is the TaqMan assay. And essentially this is again based on a very simple principle. You design a primer in a manner that you have let's say a fluorophore F1 and a fluorophore F2, and this is based again on the principle of FRET, fluorescence resonance energy transfer. Alright. The way it works is the following. Now when you have this kind of a situation that you have a primer with one end which is having a, let's say, a red fluorophore and another which is having a green fluorophore, and these two fluorophores are designed in a way that the emission from one, let's say in this case the emission from green one, is also the excitation for the other. In this case it is for the red one. Okay. So the emission spectra of this particular dye molecule, okay, the green dye molecule, is same as the excitation spectra of the red molecule. So there is an overlap between the emission of the first molecule and the excitation of the second molecule. What do you think will happen? So when there is a wavelength which comes and hits the first molecule and provides h nu energy, it starts to get excited, and it starts to emit a certain frequency, let's say nu 1, which is also good for exciting this red molecule. So essentially there is almost always a reduced green fluorescence and an increased red fluorescence, just because of the fact that you have some coupling between the energies of both these molecules. But this happens only when the molecules are placed to a certain distance which is equal to or smaller than a very fundamental physical quantity called the Forster's distance. So there is a certain amount of distance of spacing between these two molecules which can make it to workably execute a fluorescence resonance energy transfer process. This transfer of the emission signal from one into the excitation spectrum of the other is also known as fluorescence resonance energy transfer, or FRET. Okay. Or this can only happen when both molecules are close to each other by a distance equal to the Forster's distance. Okay. Forster's distance. So when they are beyond this, there is no coupling between the two molecules essentially, and so the emission from one is lost into the ambient, and then there is a certain decrease in the emission from the other, because there is a decoupling action of the excitation frequency. So essentially if you plot such a situation, let's say E1 here is the emission peak for the first molecule, and E2 is the emission peak for the second molecule. So in case there is a separation of these two molecules beyond the distance of the Forster's distance, there is a sudden growth in E1 and sudden decrease in E2. Okay, so that's what happens essentially. And as you see here, let me just clear all this out so that you can have a good idea or a good view. So essentially these different steps are indicated in this particular illustration here. You can see here that the primer comes, okay, and it kind of starts with a binding over it, separated by distance equal to Forster's distance of a green fluorophore and a red fluorophore. And during the denaturing step, the molecules separate. Then there is an annealing which happens at a certain temperature, where the primer goes and binds to one flank or one end of the DNA, and beyond that there is an enzyme which starts cleaving off whatever is there on this particular end, this end group, and starts suturing or extending the DNA molecule in this manner or in this fashion. So this is what this Taq enzyme does. So as it does that, it liberates this fluorophore and makes it free, makes it in a free molecular state in the solution. Immediately there is an increase in the green signal because it has gone away from this red molecule, and there is a dip in the excitation emission of the red molecule, whereas there is an increase in the emission of the green molecule. So this can give you an illustration of whether the PCR process is going on well, and that every step has TaqMan biting the DNA and creating this effect. Okay, so essentially this also gives a real-time snapshot of the PCR reaction. However, the problem in this case is that the instrumentation is a lot more complicated because you essentially have to find out or scan the solution at two different emission spectra and also provide two different excitation spectra, and then the spectra has to be sensitive enough to detect what is the changeover between the two emission spectra as this process happens, and this has to be done at every cycle, and there has to be a data point at every cycle. So there would be a gradual growth in the green fluorescence in the solution with a more number of copies and more number of these enzymatic termination of these labeled primers on the green level on the primer. As the reaction proceeds and the DNA grows and these enzyme steps are repeated again and again, there is a huge increase in the green fluorescence over the red fluorescence, and that gives the success of that demonstrates the success of the PCR process. So this is essentially a very interesting way of detecting whether the reaction is going on successfully. The third very interesting way is again called molecular beacons technology, and what I would like to discuss here or share here is that not all PCR assays are available with this molecular beacons. You do have to be lucky in order to get something where we can use a molecular beacon for executing the PCR. The sequence is essentially what makes a difference. And here in this particular reaction, there is a hairpin loop DNA molecule, something like this. Okay, so there is a hairpin loop structure, and by hairpin loop, what I mean is that you have certain amount of coupling, as you can see here, between the two strands. There are certain base pairs which are exact complementaries of each other, and they couple to each other. And so these lower ends or lower flanks of the DNA of the single strand DNA does bind to each other, whereas the molecules in a certain portion of the DNA over and above the ones which have already bound are essentially not complementary, and they don't bind to each other. So what happens is that there is development of this loop, and this loop is bound essentially by this bonded portion of the single stranded DNA molecule. So you have a molecule and you are essentially folding it in a manner that a certain portion here is not complementary of each other, whereas the ends here are complementary. So they kind of fold up to form something like a hairpin loop structure. Okay, something like a hairpin loop structure. Or you have these at the end flanks bonded to each other, and these as the unbonded molecules. How can we make a difference here? So essentially, if this DNA already had a reporter and a quencher molecule pretty much like a fluorescence or a fluorophore and a quencher. Quencher is something which quenches the fluorophore or it tries to draw the emission spectra of the fluorophore, and essentially there is an absorption, there is provenance of absorption over fluorescence. So a quencher with the fluorophore would mean that the quantum yield which is generated by the fluorophore is captured by the quencher and quenched essentially. So you have a lower signal, okay, if you have a lower energy signal or intensity signal of the particular emission frequency of the fluorophore, okay, or the reporter dye in this case. So you chemically immobilize the reporter at one end and the quencher at another with an understanding that as the hairpin loop is formulated, there is almost a close proximity of the reporter and the quencher molecule, as you can see here in this particular example, okay. So as the hairpin loop is folded and there is a development of these bonds, the reporter and the quencher molecules come close by, and therefore whatever is emitted by the reporter is essentially gathered by the quencher molecule. The fluorescence level, overall fluorescence level is low. Now what happens when it binds or it starts binding? So another important or interesting fact is that we can actually minimize the number of complementaries, okay. So make it just about enough to hold this hairpin loop, and if we can do that by just cleaving this and making it shorter, and this is our primer zone for a certain reaction to happen, okay. So we can actually see whether this particular sequence of bases on this hairpin loop essentially, or the loop area, is complementary to the end sequences of the target DNA, that as soon as it binds it kind of opens up, okay. So you are binding this on let's suppose, let me just rub all this off for the interest of a little more clarity, okay. So we are suppose looking into a denatured DNA, okay, and we want to find out how this strand would bind. So let's say we have a denatured single stranded DNA in the proximity of a hairpin loop like structure with a reporter and a fluorophore, okay, and let's say we have these dangling sites or bonds. So immediately as it is able to identify its counterpart, it is going to bond, and in the process of bonding it is also going to, let's say it bonds in this particular area. So the reporter and the quencher is straightened out, this hairpin loop is essentially straightened out into a full size primer, and in the way the reporter and the quencher molecules are separated by beyond a certain distance, okay. And because of the separation there is a growth in signal, there is a growth in the reporter signal because the quencher is now far away from the reporter, and essentially that is what would cause or would define the success or failure of the PCR process, okay. So essentially if the molecular beacon in question, the loop part of the hairpin loop actually goes and bonds, it kind of opens the loop up, okay. So the small amount of base pairs which are complementary, which are holding the pin part, would actually split up because the loop part is much more in size, and of course it has much more molecular forces than the few bonds which are making the pin part, and in that case essentially as it flattens out, the reporter and quencher would move away, and therefore there would be a sudden growth in the reporter signal on the whole system. So it can give again a very good real time snapshot of what step the particular real time PCR process is in, okay. So as more and more amplification would occur, the probe binds more and more, and of course there would also be a sort of TaqMan wherein whenever the enzyme comes and cleaves away the reporter molecule, it goes into the solution, and then there is no question of being near the quencher, and automatically the fluorescence signal grows with time. So essentially these are the 4 different techniques or the 4 different assays which are available for gradually monitoring fluorescence and trying to see the real time snapshot of any PCR process. Let's talk a little bit about thermal cycle. A very interesting issue here is the time of the PCR reaction, and as we have to rapidly thermally oscillate a certain fluid in its volume, almost always there is an issue related to thermal management or heat transfer across the fluid, and these some of these snapshots here taken from real time signals on microchip PCRs do show how these thermal cyclings can be rapidly varied. So this essentially is the 90-degree step. This is about 55 degrees, okay, and this is about 72 degrees Celsius step, and you're repeating this every time. In the process, you have to actually ramp the temperature down in this region and ramp it up in these regions, okay. So you have to have a proper thermal management for getting, you know, a proper thermal management for getting a better, you know, cycling time, essentially a lesser cycling time. So the ramp up and the ramp down rates are very critical to determine the overall time step that the reaction would need. Of course, there is a hold time at a certain step, because that is rate limited by other mechanisms like diffusion and enzyme kinetics, as we have been talking about before. But then if we assume that those times of hold steps at different temperatures are primarily fixed, we do have the flexibility and luxury to vary the ramp out, ramp down and ramp up rates, so that we can squeeze the cycle more and make it less time consuming. So microchip PCR essentially works in that domain, where the ramp up and down of the temperature, while it's changing between two or more temperature states, can be essentially squeezed and made almost very rapid. There are all sort of techniques which are available. There are papers where it talks about maybe a droplet PCR, where there is a small micro cantilever made up of conducting material, which is led all the way into a droplet, and there is an oil drop on the top, and this cantilever is able to heat the droplet internally at a very high rate, at a super fast speed, as a result of which PCR reaction can be accomplished in as low as about close to 2 minutes. Okay, whereas in normal cases, when you put these vial size reactions inside conventional thermocyclers with huge aluminum blocks, it would take as much as about 4 to 5 hours sometimes. So diagnostics can be made really rapid by taking care of the volume of interest, okay, volume of the sample to a smaller scale, and also by trying to go into a more surface based heating arrangement than a volume based heating arrangement. So these are some of the positive impacts that microsystems make, particularly in clinical diagnostics using PCR or polymerase chain reaction. So this essentially shows some plots here, which are signal plots acquired by a National Instrument LabVIEW package on some real-time microchips, which we have developed over the past some years, which our lab has developed over the past some years for different thermal cycling modalities. As you can see here, the hold steps really are constant. They are not varying much, but what we do vary is the ramp up, ramp down rate immensely, tremendously, okay. And so essentially this is almost reduced from 5 to about an hour or so, and better thermal management can be possible with novel means or intuitive designing. One of them I really mentioned a few minutes back. Okay, so that's about the thermal cycle. Normally we do need to read out the fluorescence signal very quickly, and for that especially in the RT-PCR or the qPCR reactions, we do have a separate reader unit. So essentially there are two aspects here. One is there has to be a reader head. You can see this is essentially an equipment from Stratogene. This is the RT-PCR equipment, okay, and this right here is also the fluorescence readout mechanism. So what this mechanism is, really it is a set of detectors which would sit on the top of these wells. There are about 96 wells in which 96 different reactions can be carried out at a single go. Okay, so these are wells, and they are covered by using an optically clean transparency with minuscule absorption. So when you are talking about an optical detection scheme, it is almost always necessary to ascertain that whatever emission spectrum is generated by the particular dye should not be absorbed by materials which come in the way of the light, okay, or on the optical path. So one of the things which do come and which is important is a cover. The cover is more important because you have to prevent thermal evaporation of the PCR fluid, and when you are talking about a small vial containing, let's say 20 or 25 microliters of a PCR solution, you are heating it in a closed confined environment to as high as about 93 to 95 degrees Celsius, there is almost a possibility that if you leave it open it will evaporate. So you do have to cover it totally with some kind of polypropylene sheet or let's say cellophane paper, where it can give a leak proof, seal proof joint, and then you can actually press the head and give a lot of pressure over these particular covered vials. At the same time, the head can be used for optical readouts. So then over every well there is actually a detector, and there is an array of detectors, and these detectors sit on the top of these small vials contained in each well, over which there is this cellophane paper, and due to which whatever is happening in each well is independently reported. These detectors are having electronic channels from 1 to 96 which record independently some of the data. Partially these channels can be just the reference without the sample to monitor the background, and partially they can actually be containing the sample. So you have a growth in fluorescence, and then you have a comparison between the reference and the signal, okay, and that way you can actually evaluate a lot about close to at least about 60 to 70 samples, maybe even a little better, about 80 samples, making few of them as references just for a proper signal analysis module, okay. And so you can actually get the real-time snapshots of about 80 reactions happening all at a single go. So it is essentially a very high throughput process. But again, because of the optics that is involved, the instrumentation which is provided is pretty expensive, and it is prone to getting damaged easily. So therefore the trend now is slowly shifting from these detectors into optical fibers. A bunch of optical fibers can do the same job even if left inside the solution, and so therefore there is a trend of making arrays or bundles, these fiber bundles, for picking up signals from these different multiple wells. And there are advanced machines which are coming up now more with this fiber technology, okay. So these are some real-time PCR results as obtained from, so this here illustrates some of the qPCR or RT-PCR data. As you can see here, these really correspond to different concentrations, initial concentrations of the template. So these are, let's say, sample 1, 2, 3, 4, 5 containing an increased order of concentration of the initial template, okay. So this is how the initial template is different in different. So the first one has the highest template concentration, and the fourth one has the least. The fifth is essentially the reference. So this is no sample, okay. So there is no DNA to start with, and there is just the other components in this reaction, and nothing get amplified and nothing increases the fluorescence, okay. And another interesting factor is the trend, as you are seeing, is essentially linear, exponential, and plateauing, okay. So there are three trends here: the linear, the exponential, and the plateauing. Why this happens is again something that I think I have spoken back in my previous lecture. So this is number of cycles, and this is, let's say, the relative fluorescence RF, okay, the growth in that. So what happens here is that in this region there is very less primer or enzyme or other molecules available, because this is diffusion limited regime. Diffusion has not fully taken place. So getting micro on this would mean that this number of cycles would really be compressed more towards the left. The exponential is really the area where you have almost all diffusion complete. Molecules are always almost all the way around the reaction, and you have started now to copy or replicate at a very fast rate. After you reach a certain cycle here, there is a plateauing phase, which means now one or more of the reactants or constituents are depleting, and because of their concentration depletion, there is a gradual reduction in the growth rate of the fluorescence. So that's why this particular trend is visible, or it comes. So one interesting factor which comes into picture while designing PCR chips is essentially how you will try to characterize the chips thermally, okay, and what you need to do to ascertain how much time it would take for the chip to cycle to a certain temperature. So let us actually draw some kind of an electrical analog for these kind of problems. We consider any thermal system to be a bunch of resistances and capacitances and circuits, okay. So let us draw this analogy one by one. So if you look at the slide here, really, you know, there is a let's say mass which you are heating up. This right here is the mass, and the mass is represented by the term m, and the specific heat of the material is C. We assume a temperature difference delta T, okay, between both ends of this mass. So delta T is the temperature difference. This may be on a higher side of temperature, and obviously because this side is a higher side of temperature, there should be a heat flow, okay, from one end to another. So there is a heat input across this end. So this is the surface with a higher temperature, and there is a heat output here from this particular surface. So this is the surface with a lower temperature, or the difference is about delta T, okay. So if we assume that Q is the heat flow, not the heat flow rate, just the amount of heat that is flowing inside the system. So Q also can be represented as mc delta T, right. So this kind of looks familiar, or it rhymes with something like capacity, right. This is called heat capacity. So essentially this is the ability to store, the ability of a material to store heat, okay, the capacity of a material, or the ability of the material to store heat. An equivalent in the electric circuit is capacitance, but there it is the capacity or the ability of a system to store charge instead of the heat. So Q in the thermal system is essentially an equivalent of charge in the electrical system, and it is very well founded that there should be analogous terms for V, the voltage in an electrical system, to the temperature in the thermal system, and also the capacitance in a electrical as opposed to the thermal system. So let us draw a capacitor. So this is a capacitor. Let's say you have a capacitance value equal to C, and you have a charge small q which is flowing through this capacitor, and essentially there is a potential difference of V here, okay. So what causes the charge really to flow is the potential difference here, right. What causes the heat here to flow is the temperature difference here. So it's a kind of analogous. So potential difference in an electrical circuit is same as temperature difference in a thermal circuit. The quantity in an electrical circuit which you flow is the charge q as opposed to the quantity in the thermal circuit, which is the heat q. And so therefore, as we know here, q is equal to C into V, the capacitance into voltage. So therefore, in the thermal circuit, q is mc delta T, where mc is the capacitance equivalent, okay, of a thermal circuit. So thermal capacity, okay, the thermal capacity of such a circuit is also mass times of the specific heat capacity of the material. To explore further, mass can also be written as the density times of area times of the depth of in this case, let us actually forget about the depth at this time. The area times of the length here and assuming that this length here is about x, okay, so it's area times of x, that is what the mass is, and so thermal capacity here should be represented as rho a x times of c, okay. That is what or how you can represent the thermal capacitance. Let us also look at if there is something analogous to a resistance in an electrical circuit as in a thermal circuit. So for doing that, we need to assume the rate of flow of charge. So basically, in case of the equivalent of resistance, what we can do is we have to consider the rate of flow of charge, okay. So if we consider the equivalent model that we drew earlier between the electrical circuit and the thermal circuit, the potential difference on an electrical circuit was corresponding to the delta T, or the temperature in the thermal circuit, and the charge Q in an electrical circuit was equivalent to the heat actually in the thermal circuit, okay. So here by the Fourier's law of heat conduction, if we really look at the heat transfer, between these two phases here and here, the dq by dt, the rate of flow of heat, is essentially represented by the K A by x times of delta T. The K here is the thermal conductivity, okay, of the material, and of course the A is the area of cross section perpendicular to the direction of the heat flow. So if this were assumed to be like a cube, so the area A is really the area of this particular face. The x is essentially the distance, okay, the distance between the two flanks of this particular mass, and delta T being the temperature difference across it. So therefore this is certainly the higher heat or higher temperature side. This is the lower temperature side, and delta T essentially is nothing but the difference between the two temperatures. So if you put a circuit equivalent, this can be more analogous to a resistance, that you have a current across it which is also the rate of flow of charge. So essentially Q dot on the electrical circuit is essentially equivalent to the heat flow rate Q dot dq by dt on the thermal circuit. And as we know here, the potential difference being V can be equated to I times of R, that is dq by dt times of R. So since the potential difference is also equivalent to our delta T here, therefore dq by dt or I is V by R, okay. So delta T is the equivalent of V, and essentially K by X is essentially same as 1 by R, or R, the thermal resistance in this case, is X by K times A. K is the thermal conductivity, A is the area of cross section perpendicular to the direction of the heat flow, and X is the length across which the heat flows, causing a temperature difference of delta T across both its surfaces. So the resistance equivalent, thermal resistance equivalent in this particular thermal circuit is X by K times of A, okay. So that in a sense the complete electrical analogy of what a thermal resistance and a thermal capacitance would be. In fact, there is an approach called bond graph modeling where these kind of concepts are very often used for kind of developing an analogy between the electrical side and the various other physical phenomena. It may be heat transfer, it may be related to optical phenomena, and there is an analogy developed. So since we have a resistance and a capacitance here, we can always draw an RC circuit and see that with the potential difference across the RC circuit, or assuming that you have a temperature difference in a thermal circuit, and a kind of series or parallel combination of the thermal resistance and the thermal capacitance, can we really develop something called the time constant? And once we develop that, the other question would be how relevant it is, or what is the physical meaning of that time constant, and can we get some inference about the system's thermal behavior, the lag or lead or the thermal response, based on what this time constant is. So let's look at preparing an equivalent RC circuit and trying to see what the time constant of an RC circuit would be. Okay, so let's suppose we have this resistance capacitance circuit connected to a potential difference, and this value is the resistance value, this is the capacitance value, and this completely closed circuit. The capacitance is C, the resistance here is R, and we assume further that subjected to the availability of the potential V, we have a current in the circuit I, which is generated because of the potential V. So V essentially is also equal to, by Kirchhoff's law, IR plus the voltage across the capacitance, which is actually Q, the charge, and let's actually write small q here in the interest of clarity, divided by the capacitance C. Okay, so that's what the voltage drop is across the capacitor, and there is a drop across the resistor, and together these drops are also the same as the voltage which is provided in the interest of conservation of energy. This is Kirchhoff's law. If you remember, we applied this law in one of the electrokinetic pumping design modules for doing circuit calculations and trying to find out the ion current as well as the flow rates. Okay, so let us solve this equation. So V essentially again is written as dq by dt R plus q by C. Current, as we know, is the rate of flow of charge, dq by dt. And so if we rearrange this equation, we are left with dq by q. And we can assume the q varies between zero charge in the capacitance at the very outset to about a full grown charge of, let's say, small q. Okay, and this essentially is this is CV minus q. This is CV minus q. Okay, so dq by dtr is CV minus q by C. Okay, and this essentially is equal to the integral of dt by RC, dt being varying between 0 and t. T is the full time over which the charge of the capacitor grows from 0 to q. So if we solve this equation, we are left with minus ln CV minus q plus ln CV. Okay, is essentially equal to T by RC. Or in other words, q here, the charge, is CV times of 1 minus e to the power of minus T by RC. All right, so CV also is a representative of the full charge on the capacitor. Let's call it QF. So Q is QF times of 1 minus e to the power of minus T by RC. Alternately, for the discharging circuit, when we assume that there is no voltage source, as you can see here in this particular illustration, the voltage of the capacitor which has been developed over its plates is going to feed the circuit with the current I, and there is a drop across the resistor. So in that case, the discharging time for the discharging process, the Q becomes QF e to the power of minus T by RC. So at time t equal to infinity, the Q essentially becomes actually 0, because this is minus. So 1 by e to the power of infinity is nothing but 0. Alternately, at time t equal to 0, the Q is actually grown to its full value QF just before the start of the discharging process. So, a very interesting aspect about these two charging and discharging cycle times are that if you plot the percentage of charge with respect to the time, okay, as you are seeing in this illustration here, this really is the growth cycle, right. So, this is essentially just give me 1 minute. So, this essentially is the way that the charging happens in the circuit following the expression Q equal to QF1 minus e to the power of minus t by RC, and this is the exponential decay in the charge Q is equal to QF e to the power of minus t by RC. So, this is the charging cycle, or this part is a discharging cycle. Also, what is interesting here to know is that if you have a time constant t of this RC, this corresponds to about 63% of the total charging. Okay. So, if you have one time constant RC, resistance into capacitance, you have about 63% of the charging, and it takes about 3 time constants for the charging to be above 95%. Okay. So, essentially we are looking at 3 time constants for the capacitor to charge to almost 96% of its full charge. This is an interesting proposition, because if we want to apply this to our PCR microchips, we may as well develop a strategy of trying to figure out how much time it would take for the ramp up ramp down mechanism of the particular chip. Okay. If I actually make an electrical equivalent or an electrical analog in terms of a thermal capacitance, thermal resistance, and a thermal potential difference, which is also the temperature, I would be able to make the electrical analogy and trying to see that what kind of time would be needed for our thermal circuit to go to a certain level, let us say about 96% of the full thermal load that the cycle can take at a particular temperature. Okay. So, in that, if we design assuming that full temperature at this value, we can obtain the full advantage of increasing the ramping rate. Now, let us kind of try to illustrate that a little more with an example. So, in this particular case, as you are seeing here, there is a chamber of PCR reactor which is made in silicon using a deep reactive ion etching process. So, this chamber is essentially a high aspect ratio chamber used or etched away using a gas plasma, the deep reactive ion etching. The chamber is a square size with an edge length of about 2 millimeters, okay, and there is a depth of D equal to 200 microns this particular chamber. So, therefore, you have a square of 2 mm into 2 mm and a depth of about 200 microns. So, the chamber is essentially pretty much high value on the length aspect and really micro in the depth aspect. Now, this is how normally micro architectures are, or they are designed. Another advantage why this almost 1000 times increase in the dimensions in the linear xy dimensions of the chamber is because the idea is to actually spread a volume of fluid over a thin surface area. So, essentially you are taking a bulk volume of the fluid. So, the idea really is to kind of spread over a thin film from a bulk, so you can increase the heat transfer. So, in this example you have to determine the minimum ramping time which is achievable, and with this particular reactor design, and you have to assume that the sample liquid is water. Most of the PCR fluid is actually an aqueous based fluid with a little bit of ionic content here and there. Okay. So, the density is around 1000 kgs per meter cube, as is normally the case with water. There is a specific heat per unit area or per unit mass of 4182 joules per kg degree Kelvin, and there is a thermal conductivity associated of K equal to 0.6 watts per Kelvin meter. And so, you have to essentially design the chamber or design this in a manner that you can determine the minimum ramping time which is achievable by this system. So let us use the theory that we have just formulated in terms of electrical analogy. So essentially we need to find out the thermal capacitance and the resistance in this case. So you have first, let's draw the chamber. You have a case where you have a 2 mm by 2 mm chamber, and essentially the depth of this chamber is about close to 200 microns, and we assume a heat transfer from bottom to top. Okay, so there is a set of heaters, let's say a set of micro heaters at the bottom of this chamber, and it is actually feeding this particular chamber. So in this case, the thermal capacitance or heat capacity let's say C thermal, essentially m times of C. M is the mass of this volume, and C is the heat capacity, okay, of the material. So essentially it is again rho times of area of cross section, which is square of A times of the depth, in this case the depth being from the heating side to the open or the atmospheric side is D. Okay, so we assume this to be A and this to be D. So you have rho square, rho A square D is the total mass, and then of course you have the specific heat C. Similarly, if you determine the thermal resistance, and that is from the heating membrane to the top of the chamber. So this is the heating membrane or the heaters, and this is the top of the chamber. So from the heating membrane to the top of the chamber. Okay, so let's call it R thermal. So in this case it would be actually X by Ka. So X essentially is D here. If you just remember the electrical analogy that we just dropped, or we just drew about a slide back, the area of cross section is A square, so we have D divided by A square K. All right, and essentially the time constant in this case, which is R thermal C thermal, is nothing but D square rho C by K. Okay, and so if we assume a D of 200 microns, 210 to the power minus 6 square times of density of water, which is about 1000 kgs per meter cube, times of specific heat capacity, which is about again 4182 joule per kg per Kelvin, divided by the K value, thermal conductivity, which is 0.6 watts per Kelvin meter, we obtain time constant of about 0.28 seconds. Right. So essentially the maximum time of response that the system should have to achieve a temperature 96% of the maximum temperature, okay, which in this case is probably 90 degrees or 94 degrees, should be 3 RC, right? As we saw in the RC example, that Q, which was essentially the electrical equivalent of the temperature, sorry, the heat. So the Q, which was essentially an electrical equivalent of the heat, big Q in our case, or capital Q in our case, was about 63% of the full charge once the time reached one time constant, and was about 96% of the full charge as it reached about 3 times RC. Okay, so the maximum time that you can allow for the system to ramp up to this particular 96% of maximum temperature in our case is about 3 times of 0.28 seconds, which is about 0.84 seconds. So the take home message here really is that the time constant is really a function of reactor volume, okay? And so if you can make the volume smaller and smaller, you could make the time constant smaller and smaller, and the volume in turn depends on the dimensions. So effectively, at this scale, the constant is independent of the temperature. So that's exactly what we have to consider for designing microsystems. So would like to finish this lecture just by trying to just reiterate that at times we need to probably draw these kind of analogies to get a quick solution of what would be the maximum time that we can allow for a system to go to a certain temperature state by going through a ramp up cycle, okay? So in the next lecture we'll take on to a little more details of how do we really design the volume based on such time constants, and then look into some other aspects of how the DNA polymerase works on DNA molecules and tries to extend a zipped up or a zipped down molecule, make copies inside the human body. We also like to kind of review a basic electrokinetic technique, electrophoresis, which is also used for finding out the length of a DNA base pair, and is critical to diagnostics industry. So I would like to close this lecture with this note. Thank you.